Hello, my name is Albany Pardo, and today I'll be talking about the transforming growth factor beta, which is also referred to as the TGF-Beta pathway. So I just wanted to make a note here. This pathway is responsible in creating the formation of axons during embryo development in the brain and for the formation of neuronal polarity when developing. Irregularities of this TGF-Beta, as in, for example, lacking the quantity of receptors needed, can cause neurons not to migrate to the needed areas of the brain that are developing. Mutations in the TGF-beta receptors have caused developmental disorders within the brain. So here on our next slide, I'm going to go ahead and include the items that will be present within the explanation of the TGF-beta pathway. So the purple spiral is going to go ahead and represent the receptor type 2. Green spiral will represent receptor type 1. The yellow circle will represent the TGF-beta ligand. The P will represent phosphates and the occurring phosphorylation that will happen within the pathway, which we will speak about later. The blue-green circle will represent SMAD2/3. The pink circle will represent SMAD4. The pink region of receptor type 2 will be the GS box, and the TF would be the transcriptional factor. So before we start talking about the TGF-B pathway in detail, I want to go ahead and talk about why this is important. So here in this slide, we have a hematopoietic stem cell here. For this example, we have this happening within the embryonic development. So upon the TGF-beta pathway happening to this hematopoietic stem cell, we would have differentiation occurring, which will result in this neuron that we see right here. This neuron is a fully formed neuron, differentiated. We have the myelin sheath, the dendrites present, and this is very important because it just shows how everything comes together. So let's go ahead and talk about the TGF-beta pathway here. Okay. So here we have in green the receptor type 1, and purple we have receptor type 2. So what we're going to be having is the ligand will be binding to receptor type 2. There we go. As a result of the binding that happens, we will have dimerization occurring within the receptor type 1 and receptor type 2. Upon that happening, we're going to have phosphorylation occurring into the GS box. Simultaneously, we would have phosphorylation occurring at the tails of both of the receptors. We have that happening here. And as a result, this will cause activation into the pathway. So this is just a side view of what's going on, just because I wanted more space to explain what is going on. So to your right bottom corner, we're going to see the double helix there. That would signify the DNA, which would be within the nucleus, which is what we see right there. And okay, so we have phosphorylation happening at the tails. Then what we have is the SMAD2/3, which will then become phosphorylated. After it's phosphorylated, it will go ahead and combine with SMAD4, which you'll see right there. So now this complex is ready for translocation into the nucleus, which we will see right here. And this complex will go ahead and use the transcription factor to bind to the promoter of the specific gene. So we would have transcription happening, which we would see right there. Now, I can go in depth into this pathway, but I really wanted to focus into a specific part that was very interesting to me, which would be SMAD7. So upon the transcription, we're going to have the creation of SMAD7 as a protein. This protein is actually very necessary into the inhibition of the pathway. Therefore, it just provides some sense of control, which is very necessary, especially within every aspect of the cell. Okay, so this is very simplified, but you might want to ask, why is this important? Well, The TGF-beta pathway contributes to the differentiation of neural stem cells during embryonic development in the nervous system, especially by promoting oligodendrogenesis and myelination, which is what we saw in the first example that I provided with the axon that was beautifully placed there. We have it placing a pivotal role in the stem cell determination within the central nervous system. This can also be observed within the peripheral nervous system as well. And to provide more insight into this, I did a bit more research to just provide more information as to why this is significant, why this is important. We have here a study that suggested that mice that had the SMAD4 knocked out, so that was no longer present within the TGF-beta pathway, inhibiting the entire pathway. We showed, the study saw that the mice exhibited schizophrenia-like phenotypes, which provided significant evidence between the disruption of the TGF-beta pathway and psychiatric disorders. Just provides us a little bit of clue of what might actually be going on. So my take on this is that we could further our understanding on the previously mentioned study by utilizing humanized mouse models while also knocking out the SMAD4 gene to accurately compare and assess the association of the TGF-beta pathway and the role of its disruption, which would cause psychiatric disorders, or at least a claim that is being made in the previous study. It would also provide a good understanding about what's going on as we're using a humanized mouse model, which would be a much more accurate way to completely understand what is really going on. Our further understanding would allow us to understand the disorders in a much clearer way and hopefully, generate better treatments that would provide a better quality of life for patients. So at the end here, I have my references that I used, and that's it. Thank you for your time.